Transcription begins with a sequence of DNA, which is comprised of a promoter followed by its corresponding gene. The promoter contains a specific nucleotide sequence called a data box, to which transcription factors combine. These transcription factors allow RNA polymerase to attach to the DNA strand and begin transcribing it. The RNA polymerase moves in the 3' to 5' direction of the template strand and produces the mRNA sequence behind it. Inside of the RNA polymerase, the two DNA strands are unwound, and the polymerase molecule assembles free-floating RNA nucleotides in the 5' to 3' direction, complementary to the template strand. Once the entire gene is transcribed, it is released from the RNA polymerase enzyme to be processed. The first step in processing is the addition of a 5' cap, which is a modified guanine nucleotide and is added, obviously, to the 5' end. Poly-A tail is then added to the 3' end, which is a very long sequence of adenine nucleotides that prevents the RNA transcript from degrading. The next and most important step is splicing. The mRNA transcript of a gene contains both coding segments, called exons, and non-coding segments, called introns. The introns must be removed before the mRNA transcript can be translated. This is done by spliceosomes. Spliceosomes are complexes of small nuclear RNA, which helps facilitate the breaking and rebinding of the bonds in the transcript and various proteins. Spliceosomes are also ribozymes because it is the RNA within them that is carrying out their specified function. The spliceosomes bind to specific marker sequences at the beginning and end of the intron segments of the transcript. They then connect together, making a loop out of the intron, and form new bonds between the now adjacent exons. The introns are cleaved from the sequence, and the spliceosomes move away. The introns degrade back down into nucleotides to be reused later. The now mature mRNA transcript has finished processing and exits the nucleus via a nuclear pore to be translated into a polypeptide at a ribosome.